Haemoglobin electrophoresis is a laboratory test. It helps identify different types of hemoglobin. This is important because hemoglobin carries oxygen throughout the body. Abnormal hemoglobin can cause health problems. This test is useful for diagnosing blood disorders. These disorders can be haemoglobinopathies like sickle cell disease. They can also be thalassemias, which are characterized by reduced hemoglobin production. Electrophoresis works by separating proteins. It does this based on their electrical charge. Different hemoglobin types have different charges. This allows us to see them as distinct bands on a gel. By analyzing these bands, doctors can diagnose and monitor various conditions. Accurate diagnosis guides treatment and improves patient outcomes. Hemoglobin is a protein. It is made up of amino acids. The sequence of these amino acids determines its electrical charge. Electrophoresis uses an electric field. This field is applied to a gel. The gel contains hemoglobin samples. Because different hemoglobin types have different charges, they move at different speeds in the electric field. This difference in speed causes them to separate. Hemoglobin variants with a more negative charge move faster towards the positive electrode. Variants with a more positive charge move slower or even towards the negative electrode. Alkaline cellulose acetate electrophoresis is a common method. It separates hemoglobin variants. This method uses a cellulose acetate membrane. The membrane acts as a support medium. First, blood samples are collected and prepared. Red blood cells are lysed, releasing their hemoglobin. Next, the hemoglobin samples are applied to the cellulose acetate membrane. Then, an electric current is passed through the membrane. The hemoglobin variants migrate across the membrane at different rates. This is due to their varying charges. After a certain time, the electrophoresis is stopped. The membrane is stained. This makes the separated hemoglobin bands visible. Section 4. Limitations of alkaline cellulose acetate electrophoresis. While alkaline cellulose acetate electrophoresis is widely used, it has limitations. Firstly, it may not resolve all hemoglobin variants. Some variants have very similar charges. This can lead to overlapping bands. Secondly, this method requires a fresh blood sample. The sample must be properly stored. This is to prevent protein degradation. Thirdly, the technique can be labor-intensive. It requires technical expertise to perform accurately. Due to these limitations, other techniques may be employed. High-performance liquid chromatography, or HPLC, is one such alternative. It offers higher resolution and automation. However, it is more expensive. Section 5. Interpreting electrophoresis results. After electrophoresis, the separated hemoglobin bands are analyzed. Their positions are compared to those of known controls. Normal adult hemoglobin, or HbA, migrates a specific distance. It creates a distinct band. Other hemoglobin variants, such as HbS, found in sickle cell disease, or HbC, migrate differently. They form bands at different positions. By comparing the patient's bands to the control bands, technicians can identify the presence of abnormal hemoglobins. The intensity of the bands reflects the amount of each hemoglobin variant present. Densitometry can quantify the proportions of different hemoglobins. This helps in diagnosing hemoglobin disorders. Section 6. Conclusion: the enduring importance of electrophoresis. Hemoglobin. Electrophoresis is a cornerstone of hematology. It aids in diagnosing and managing blood disorders. By separating hemoglobin variants based on their charge, this technique allows for the identification of abnormalities. Despite limitations, alkaline cellulose acetate electrophoresis remains a valuable tool. It is accessible and relatively inexpensive. However, evolving technologies like HPLC offer enhanced resolution and automation. As our understanding of haemoglobinopathies advances, so too will the techniques used to study them. Yet the fundamental principles of electrophoresis will undoubtedly remain relevant. This technique will continue to guide clinical decisions and improve patient care for years to come.